So this chapter is about viruses. So the first thing is, what are viruses? So viruses are, most of the time they're considered non-living. So they're just made up of DNA or a nucleic acid and protein, and they have to have a host in order to reproduce. So the big thing is they're ubiquitous. They're everywhere. They infect everything that every living thing that we know about. Anything that's living that we know about, we've found a virus in them. They found a virus in them. So things that live in the ocean, everything has a virus in it. Algae, bacteria. They are of like historical significance. So the big flu pandemic of 1918 caused more deaths than World War I. So it actually killed 500,000 Americans in 10 months. So I posted a video here that you can watch about it. And then, there's actually lots of videos. Some of them are more detailed than others. But I just want you to get the idea how a pandemic can spread. So pandemic is an epidemic, but a pandemic is worldwide. Okay, and then so it's been 100 years since the last big pandemic. So they had a commemoration, not really a celebration, but a commemoration of the 1918 with the CDC. So both of those links have some information about it that you guys can read. Okay, the history of viruses. So Beijerinck, he was one of the first people to propose that a virus was some sort of infectious fluid. So they were trying to figure out what was causing certain symptoms. And they had a filter that kind of looked like this, a ceramic filter that would actually filter out bacteria. But what they got out of it, the liquid that they got out of it, still caused disease in plants. So they were like, well, it's got to be something smaller than bacteria that's causing the problem. So, so they're saying, oh, it must be non-living, but it's infectious fluid. So how can that be? One of the first viruses that they found was tobacco mosaic virus. So it's like the first one. And if you look, here's a plant with symptoms of it. It causes parts of the plant tissue to yellow. It's pretty economically significant to tobacco farmers. So a lot of money has gone into it because of tobacco industry. So it's one of the model viruses that they use to study and learn how viruses actually replicate, what they're made out of, and that kind of thing. So that's TMV. It's really, really contagious between plants, not people, just plants. Okay, so in 1884, Pasteur's the one that came up with the term virus. So viruses like venomous or slimy fluid, so it's a poison. One of the first human viruses that was demonstrated to be a virus was yellow fever, and Walter Reed was the person behind that research. Finley was the one that actually proved it. It was also kind of complicated because they had to prove that it was some sort of infectious agent that was actually transmitted by mosquitoes. So this was going on when they were working on the Panama Canal. Okay, so viral components, so they have a nucleic acid and they have protein. So the function of nucleic acid is always for coding for protein. Okay, so that's why we have it, is to make protein. So the virus is going to use the host machinery, so the host ribosomes, to make protein that it needs. Okay, so the nucleic acids can be either RNA or DNA. So virus could start with RNA, doesn't have to be DNA. So that's kind of an exception to everything else we've learned. It could be single-stranded DNA, single-stranded RNA, or it can be double-stranded. Okay, here's the picture. So here's the nucleic acid in here. Around it is what's called the capsid, and it looks purple on this picture. So the capsid is protein, okay, and it's made up of these little subunits that are called capsomeres. So capsomeres, I kind of think of them as Legos, but somebody said, oh, Legos aren't circles, but but they go together. They kind of link together. So it's like little building blocks that you can put together. And they make up the capsid. So the capsid's going to determine the shape of the virus. They can be used for attachment to a host cell. Okay, a virus particle that's assembled is called a virion. This virus has an envelope and it has spikes, which I have on the next slide. Okay, so not every virus has an envelope or spikes, so they can just have the nucleic acid and the capsids. Okay, so the envelope is the outside layer, and it's made out of lipids. Okay, so it's a lipid bilayer. They can have these proteins sticking out of it. So it's extensions from the envelope. They can be glycoproteins. So the spikes can actually serve as points of attachment for the virus to the host. So these little spikes might attach to the host cell. So if this is a host, okay, and this virus comes up, it can attach here. The envelope, everything doesn't have an envelope, so don't necessarily have an envelope. I have a link to this HHMI video. It's about HIV, and it shows HIV, which is a virus with the envelope and spikes that we know a lot about, and it explains how it infects. I'm not worried that you memorize the detailed names of the spikes and the receptors and everything, but I want you to see, in general, I want you to watch it so you can see how the how HIV infects, and you can see how the parts actually work together. It explains it really, really well. Okay, so the advantage of having an envelope is it actually can mask the virus from the host immune system, so it kind of looks like our own cells because they have plasma membrane. It can assist in infecting healthy cells because, whoops, sorry, wait, the cat's up here laying on my laptop. Here's, if you think about endocytosis, so here's a host cell again. Okay, here's a virus with an envelope. It can come up and merge with this, and it can become part of it, just like in the HIV video. The disadvantage is the viruses with the envelope are not as not as tough. The envelope, the lipids, can actually be damaged by heat, which I have temperature. That's the same thing, right? pH, chemical disinfectants can destroy them easier than if it's just a protein. Okay, so now you can hear the cat purring. Okay, so a naked virus only has a capsid and no envelope. Okay, so no envelope. Mia. They're way more resistant to harsh environments, so they're tougher. They're harder to disinfect. Viruses, viral shapes and sizes. I don't expect you to memorize these, but I just kind of want you to see how diverse viruses can be and their relative size. So here's a human liver cell is what this brown is supposed to be, and then within it is this green cell, which is supposed to be E. coli. Around it or within it, you can see different shapes. So pox viruses, those are things that cause chicken pox, bacteriophages, which we're going to talk about, TMVs in there, herpes. Okay, so just a bunch of different things. And then here's some pretty colorized pictures from the scanning electron microscope. Actually, it's probably TEM that they colorized, so you can see what the viruses look like. So there's phage DNA, or phage, not DNA, phage virus, and there's TMV, and then there's HIV. Okay, so here's more information about the shapes and sizes. So one of the biggest viruses is smallpox. It kind of looks like a pine cone. And one of the smaller viruses is polio. And you don't have to memorize those. The shapes are determined by the capsomere or the envelope. And there's all sorts of different shapes. One of my favorite ones, this is Ebola, and it's a filament. Some people say they can see Mickey Mouse in there. I don't know. Oops, now she took the mouse. Okay, host range and specificity. So viruses are extremely specific to what they can affect. So the viral host range are different kinds of hosts that a virus can infect. So like, can it infect people? Can it infect animals? Okay, so most viruses are limited to one host. Okay, they can only affect humans. So like polio infects humans. Rabies, okay, can go from species to species, right? So it can be in dogs, it could be in cats, it could be in raccoons, it can be in bats, okay, and then it can be transmitted to humans. Okay, now viral specificity is what kinds of cells it can infect. Okay, so it really depends on the receptors that are on the cell. So viruses can be very, very specific to what cell type, so whether it's epithelial tissue or connective tissue, okay, or nervous tissue. Okay, so papillomavirus. So papillomavirus is causes warts, or HPV, right? Can cause genital warts and can lead to cancer. So it infects epithelial tissue. Okay, hepatitis is going to infect liver cells. Okay, and then this cytomegalovirus, it's actually an example of one that can infect diverse cell types. So it can infect salivary glands, it can infect your GI tract, which salivary glands is there, so liver, lungs, and it can actually cross the placenta if you're pregnant and infect the fetus. Okay, so host range is the types of organisms, and then specificity is types of cells. Okay, so virus classification. I just want you to realize that viruses aren't classified the same way that everything else is. Okay, so we're not using Linnaean classification to describe viruses. Viruses, because they're not considered living, they kind of have their own system, and it's the International Committee of Taxonomy on Viruses that meets every couple years and comes up with how they're going to name them. There's a lot of different viruses, so at least 5,000. They usually use an English name rather than a genus and a species in Latin. How they're classified and how they're organized is based on the nucleic acid type and what structure it is, maybe how it replicates, what it can infect, what kind of symptoms it causes, so other chemical and physical characteristics. So usually a lot of them are based on what kind of symptom it causes. Okay, now, one thing I want you to remember, okay, so when we talk about the central dogma in biology, we always go from DNA to RNA to protein, right? And so this is transcription, right? And this is translation. So in viruses, right, if we have a DNA virus, we're going to go this way. So DNA to RNA to protein. Now, if we have a virus that's just starting out to be RNA, right, we can go straight this way. Okay, or we can have it where we actually copy it and make another piece of RNA and go this way. Okay, or we can have a retrovirus, which is like HIV, and a retrovirus, so I'm going to make retro orange or yellow. A retrovirus would go this way. And so this is retro. In a retrovirus, we use the enzyme reverse transcriptase. Okay, so what we're going to do is we're going to do the opposite of the central dogma. We're going to take RNA, okay, and we're going to copy it and make DNA using a different enzyme. So the only things that do that we know of right now are retroviruses, so HIV. So it's exception to the central dogma of biology. Okay, so now a bacteriophage. So bacteriophages have been studied a lot, so that's another model organism. What they are they're viruses that infect bacteria. Okay, and you can look up, I meant to put a link in here and I didn't. You can look up phage therapy. So I'll put a link on Moodle for you. So phage therapy is the idea that if these viruses infect specific strains of bacteria, phage therapy is the idea that maybe we could give people viruses, these phages, instead of antibiotics. Okay, so an example is a phage they call T4. Okay, so T4 phage actually infects E. coli, and this is a picture of it down here. So here's the E. coli and all these little yellow things on it that are colored yellow. Okay, those are bacteriophages. So they can go in there and they can kill the bacteria. They're very, very host specific. So if the host dies off, the virus doesn't have any place to go, right? The virus doesn't have any place to go, and the host, in theory, the virus is going to die. Yeah, the FDA hasn't approved it yet. Okay, so we don't use this in the United States. There are some examples of other countries where it's used. So there's a phage institute in Georgia, which used to be part of the Soviet Union, Georgia, not our Georgia in the United States. But in Georgia, they have a phage institute where they research it and they use it to treat infectious disease. Okay, there's more research going on in it. I'll post some links so you guys can think about it. Okay, one way that we can screen for phages is actually this thing called a plaque assay. So a plaque assay, what you do is you take a petri dish and you cover it with bacteria, and then you put some water on it. It could be water with soil, and you put it on here, and wherever you get a clear zone, okay, that's where a virus is that's actually broken down the bacteria, so it's eating the bacteria. So all these clear areas are where the phages are, and you can find them everywhere. They're everywhere. Okay, so here's the anatomy of a phage. Okay, so here's the envelope. Okay, and there's where the DNA is. Actually, it's probably not the envelope, it's a capsid. Okay, and then it's got a sheath and a base plate, and then it's got these little legs, okay, or tail fibers, and these are going to attach to the organism. Okay, now there are 5 main steps in how a virus replicates. Okay, so there's adsorption. Okay, so if we go here, so absorption is when the virus is absorbed into the bacterial cell wall. Okay, so those little feet, I guess we could go back. Okay, so absorption is the phage is adsorbed onto the bacterial cell wall. Okay, now this is the circle is the bacterial chromosome. Okay, now penetration, what happens is the phage penetrates the bacterial cell wall membrane, and the DNA actually just kind of gets injected into the bacterium. Okay, then the bacterial DNA is disrupted. Okay, and biosynthesis occurs. Okay, so biosynthesis is when the phage DNA kind of takes over the cell, and it tells the cell to make the virus's proteins and the virus DNA. So that's biosynthesis. Okay, and then maturation, what will happen is we'll take the baby phages and we'll put them together. Okay, so the collar sheaths and base plates have been attached to the heads. The tail fibers are added last. Okay, so we're making new phages. And then release, what happens is the bacterial cell will die, right? And the mature phages are released and they're spread out. Okay, and then they can absorb to other cells. So those are the 5 steps of viral replication in general that occur. Okay, there's variations on it, but this is in general. So absorption, penetration, biosynthesis, maturation, and release. Okay, now I have a video on here where she explains it on Khan Academy. So bacteriophages have two cycles. They have two ways they could go. So we could have a lytic cycle, or we can have a lysogenic cycle. Okay, so they can follow that normal 5-step thing, but then they have some variation. So I want you to watch her video where she explains it, because she does a really nice job and she draws out the pictures nicely compared to what I can do. Okay, so here's the lytic cycle. Okay, so in the lytic cycle, what we're doing is we're making new phages. In the lysogenic cycle, what's going to happen is the phage DNA is going to become part of the bacterial DNA. Okay, so the phage DNA gets integrated. So it becomes what we call prophage. Okay, and they can hang out like that indefinitely. Okay, and she explains how it all goes on, so click that link. Okay, now, the prophage is important because, okay, so if this is our bacterial DNA, right, and here's our phage DNA. Okay, so this is the bacterial. Okay, and this is the phage. Okay, and this thing is called a prophage. What can actually happen is the bacteria will make the protein for the phage, and that protein can be a toxin. Okay, so some bacteria are only pathogenic if they have if they're infected with the phage. They have the prophage. Okay, and that makes a toxin. So some examples of these bacteria, one is diphtheria. There are some vibrios. I think there's one more, I can't remember. Let's see with my notes. Cholera. Oh, Vibrio is a cholera. Vibrio causes cholera. Okay, so these organisms aren't pathogenic, okay, don't cause disease unless they've been infected with the virus. Okay, now I'm going to stop here and upload this, and then I'll make another set of videos for the last 7 slides.